Hello and welcome to microbiology. We're going to be using Kelly Cowan's Microbiology, a Systems Approach, 5th edition. With these narrated PowerPoints, all you really have to do is sit back and listen and allow me to talk to you about microbiology. The first thing we should probably do is talk about really what microbiology is. Well, microbiology is the study of small living things, hence the name microbiology. And so whenever we talk about these organisms, there's some appropriate terms to use. Things like microorganism or microbe works. A looser kind of example is something like germs. But really I prefer more specific things. So if we're talking about a virus, let's call it a virus. Or if we want to be general for anything that could cause infection, we can call it an agent. One term I don't like is bugs, because bugs are considered to be insects or arachnids and not microbes. So whenever we talk about microorganisms, we're really going to group them into really 5 major categories: bacteria, algae, protozoa, humus, and fungi. Bacteria are always going to be single-celled. Algae can be single or multicellular. Protozoa should always be single-celled. Humus should be always multicellular. And then fungi can be single or multicellular. The point is, really, pathogens occupy almost every one of these groups, with one exception, and as algae. So as important as algae actually is, because they're huge photosynthesizers in the aquatic environment, unfortunately they don't make a lot of people sick, and we really take that skew that's a medical style microbiology. So we won't talk much about algae really at all. Viruses are another important thing, but note, viruses are not even cells, and really, not even being a cell, you can't even be considered alive. So it's important to understand that viruses are non-living microbes that can make people sick. So whenever we talk about really the scope of microbiology, it's important to understand that there are blessings and curses here. And one of the blessings here is microbes actually reproduce very quickly. You can have a whole new generation of E. coli within 20 minutes. While if you're studying something like rodents, even though they're quick in terms of animals, it's still going to be many, many days to months. And so the idea here is we can grow these microbes quickly. One of the other pluses here is, since they're not considered animals or even vertebrates here, no one really cares much what we do to microbes. People don't get offended if we actually kill a bunch of microbes. While if we were to kill a bunch of rodents or cats or whatever else, people would get upset. And so one of the beauties of working in microbiology is you can manipulate the microbes and typically not draw really ramifications from the public. One of the bigger things I want to point out here that's kind of a curse is the fact that we can't see these organisms without the aid of a microscope. And so it's important to understand that whenever you do your work, you're not going to be able to see what you're doing. And as a result, you don't know if you screwed up, or if you've contaminated something, or if you're actually carrying microbes in places that you shouldn't be. So again, being safe and smart is very crucial here. The other negative here is these things can be pathogenic or dangerous to us. And so the idea here is you should treat all microbes as potentially a threat until you know better. So again, there are pluses and minuses, and there's something that you kind of have to get used to versus other types of biology. So what do microbiologists study? Well, they study a variety of things. We look at things like cell structure and function, growth and physiology, genetics, taxonomy and evolutionary history, even interactions with the living and non-living environment. But it's important for you to understand that typically a microbiologist will have background in all these different areas, but will typically focus on one specific topic. For me, myself, I would be what you would call an environmental microbial geneticist. So, as you can imagine, we are going to have different branches or areas of microbiology, but I want to stress some of these are more relevant for our class than others. Like for us, medical microbiology is quite the important topic, or even public health and epidemiology. So these are areas that we'll cover pretty extensively. Another area that we'll look at is immunology. Immunology is important in really protecting against infection, which is a big part of what we'll actually do in this class. But there are lots of other areas of microbiology that we won't get to study, although they're incredibly important. Things like industrial microbiology. Believe it or not, a large amount of goods that you actually utilize can and will be produced using microbes, including things like a lot of foods, like your dairy products, like your yogurts, and even things like beers, drugs, or even enzymes. Another area that is also incredibly important is agricultural microbiology. Believe it or not, a huge amount of our ability to grow plants and do agriculture relies specifically on microbes. And so understanding how those microbes interact with agricultural products is very helpful in us producing the max yield that we possibly can. One other place that I have a rather big fondness for is environmental microbiology. Again, I did work in environmental microbiology, but again, with this class being medically focused, unfortunately, we won't get to spend much time on discussing how important microbes to actually our environment that we live in. One thing I do want to stress before we move on here is it's important to understand that microbes are absolutely necessary for our survival. Without them, we could not live. So looking in things like agricultural and environmental, we really get to see that interaction. We'll kind of hint at it as we go along, but I really want to kind of just bring it to the front of your attention for just a minute there. So the next thing we should really talk about is the impact of microbes on Earth. This understanding that, again, microbes have been here for a very long time and actually really shaped the development of everything else on this planet. Again, we think they popped up around 3.5 billion years ago and really lived by themselves, creating an environment that's much more favorable for other forms of life. I do want to point out, though, again, we do think these were the original inhabitants, and as such, every other thing has developed from them. And so, again, they're the beginning of our evolutionary history. And it's important to understand that really from that unknown previous ancestral cell, we've seen the three major domains, bacteria, archaea, and eukaryotes, actually coming from that last common ancestor. So naturally, we're going to have to do a little talking about the different categories tied to kind of taxonomy. This is not official taxonomy here. Again, officially we have three domains: bacteria, archaea, and eukarya. But I want to point out that eukaryotes are the most different out of those three, and they're their own unique group, again, that we call eukaryotes. But prokaryotes are actually made up of both bacteria and archaea. So bacteria and archaea are going to be different enough to be separate domains, but have enough similar features that they're clearly unique and different from eukaryotes. And one of the bigger differences here is the fact that prokaryotes do not have a nucleus. So to make this super simple, if I'm talking about something that has a nucleus, it has to be eukaryotic. So everything like plants and animals and insects and fungi and protozoa, those are all eukaryotes. The only things that are prokaryotic are bacteria and archaea. So whenever we actually kind of look at this timeline, I want to kind of point out some things to you. First off, I want to stress, notice we think that life has really popped up around this 3.5 billion years ago. This is that previous ancestral cell. We don't know what it was. But from there, notice again, we do see the three domains showing up coming from that ancestral cell. But what's actually more interesting here is note how everything we can think of today is actually really not that old. In fact, insects popped up much earlier than we did, or even reptiles, mammals. But the other thing here is note how small the bar for humans is. It's basically non-existent. But one thing I want to stress to you is think of the impact that we've had on this environment for the short time that we've existed. So let's point out, microbes are a word that we call ubiquitous. So ubiquitous means to be found everywhere. Okay, so if you hear the word ubiquitous, that means it's found everywhere. And really, whenever I say everywhere, we're talking anywhere that we see life, we see microbes. So deep in the Earth's crust, polar ice caps, oceans, inside of ourselves, and even plants and animals, right? So this idea that microbes are found literally everywhere. And so really, we can see a huge importance on their activities in the environment. So we were kind of teasing about evolution before in terms of just discussing it, but we should probably actually really go into it. In fact, the theory of evolution is a very important part of biology overall. I do want to stress this is still a theory, but again, theories are held based off of the scientific method at pretty high esteem, and it's important to understand that really we hold evolution to the same level we hold cell theory, or again, homeostasis, or even genetics. So the idea is evolution is very well supported. Again, you can agree or disagree all you want, but do understand evolution is scientifically pretty sound. And it's actually pretty simple. I do want to point out that evolution in no way describes where life came from. We don't actually know that yet, nor will we probably ever figure that out. What's more important to understand is evolution is basically things change over time. That's really what evolution means. And that's not far-fetched. Think about basically every aspect of your life. The reality is things change over time. And you either adapt or you die. If you adapt, that change is helped pushed on for future generations. And that's what we're talking about. People are quick to ask, well, why don't we see humans changing? And the reality is it takes large amounts of time for evolution to become evident. We're talking multiple generations. We're talking thousands. And the fact is the generation time between humans and bacteria is pretty different. So let's just say for simple math that the average generation time for a human is 20 years. So it takes 20 years before you have your first offspring. And it would take 20 years for that offspring to have their next offspring, and so on. So really over the course of 100 years, which is loosely around how long we've been really seriously discussing evolution, we've only really seen 5 to max 10 generations that could have popped up. But we just said it takes thousands of generations. And so the idea here is that really we haven't had enough time to observe humans, to observe true evolution. But one cool thing about microbes is the generation time is significantly different. In fact, I talked about just a minute ago this idea that E. coli can reproduce in as short a time as 20 minutes. So really, if we were wanting to look at, 5 generations of E. coli, we're literally talking about 100 minutes. That's not very long. That's a little under 2 hours. And so the idea here is we don't really see it in people, but we're starting to see it show up in small ways in the microbial world. And so again, we see support for evolution. And again, this is not in any way an explanation of the origin of life. So let's talk about how, again, microbes have had such a big impact on our planet. As I said before, again, it's important to understand that really single-celled organisms have developed what is the world's ecosystem. They're primary producers. They actually produce more oxygen than any other thing on this planet. In fact, photosynthetic microorganisms like bacteria and algae actually account for more than 70% of all photosynthesis and thus contribute the vast majority of oxygen. I'm not saying that we should go cut down all the trees or anything. I mean, they're still really helpful, but I'm saying really if we lost all the microbes like bacteria and algae, we would be far more screwed than if we lost all the trees. So an example here is the pond with algae. Algae's photosynthetic microbes. And believe it or not, since the majority of the planet is water, there's far more algae than there will ever be plants. Another big impact the microbes have on our planet is decomposition. Do you understand we have a finite set of resources on this planet? We only have so much carbon in existence here. There's only so much nitrogen or gold or whatever other element. And so this idea is for life to continue, we need to break down pre-existing life, and that's what decomposition is. And really the two things that carry out decomposition are bacteria and fungi. So without them, we couldn't live. In fact, nothing could live. So the reality is decomposition is just another area that illustrates how important the microbes are to even our existence. Microbes are going to be also very important in forming things like soil. Again, soil is developed through decomposition of organic material. So without them, we wouldn't have soil. There would be no dirt. Again, the breakdown of rock even. That stuff doesn't happen if we don't have microbes. So it's very, very, very crucial to understand that we need microbes. Without microorganisms, we literally could not be alive. So the other area here that I should emphasize before we move on is pretty important. And it's this idea that most living things have interacted with long enough over time that they are now somewhat reliant on it. Many plants straight up require a cohabitation of bacteria to allow them to function effectively. And even us. We actually require microbes in and on us to keep us healthy. There are lots of examples where failure to have the right microbes in the right place actually results in disease. A good example is what people refer to as C. diff. C. diff is an overaccumulation of the bad bacteria, Clostridium difficile. And so the idea here is whenever we don't have the right bacteria in the right place, bad things can grow. and the result could even be disease. So the important concept here is we need microbes in and on us. But again, the right microbes in the right places. So one thing I want to stress is this word, and we'll formally talk about it more later, but I want to bring it up, and that is the microbiome, or even what we call the normal flora. Okay, so this is two words basically mean the same thing. The microbes that live in and on us that are actually really an important part of us. So really, besides using microbes in and on us, we've actually used them to do a lot of activities for us. So we've been harnessing microbes, whether we knew it or not, for a very long time. The fact that we were able to brew beer and wine and make bread well before we ever discovered what was the actual cause of this was very, very important. Again, fungi are usually used in things like cheese productions, and even this example of Egyptians using moldy bread to treat wounds. They didn't know it at the time, but really they were using antibiotics. And so the reality is we've used microbes for really an incredibly long time, thousands of years, but we didn't even know they existed. So to give you kind of a general idea here, you were more likely to give your child beer in ancient Egypt than water. So because really beer you could assume was safe to drink, while water could have pathogens that made you sick. So we were smart enough to make beer, but not smart enough to boil our water before consuming it. And so it's important for you to understand that, again, they didn't know what they were doing. They didn't understand. They kind of thought of it as almost being magic. And so the idea here is we've used them, they've been around, we've interacted for a long time, but again, purposeful use really wasn't there. Where we actually talk about purposeful manipulation of microorganisms to produce products, we really talk about biotechnology. So again, the Egyptian moldy bread thing, that's not biotechnology. That was accidental use. And so the reality is biotechnology is purposeful manipulation. And one thing we can actually do to increase overall production is something called genetic engineering. So I want to point something out to you for a second. Notice at the bottom here we have this word genetically modified organism, right? The reality is anything that has different genes or genes that have been altered through some act is considered to be genetically modified. And for some reason in our society, genetic modified GMOs or genetically modified organisms are in this huge focal point right now as being a very negative thing. The fact is genetic modification of organisms has really existed since animal husbandry, really since agriculture started. You don't take your best cattle and breed them with your worst cattle. Instead, you want to manipulate genes by breeding good cattle with good cattle to produce hopefully better cattle. That in itself is actually genetic modification. So why suddenly GMOs are going to destroy the world, I really don't understand. But the point here is, again, what people are actually afraid of is something called recombinant DNA technology. This is where we're going to move genes from one organism to another one. And this is what people actually think of whenever you say GMO. But they, again, don't understand really what they're talking about. So the reality is, beer is actually around recombinant DNA technology. This really brings me to an important point, though. Why would scientists spend all this time and effort to try to kill you? In fact, what has happened through science and technology over the last 100 years? It hasn't been an increase in deaths. It's actually been an increase in life expectancy. So if scientists are out here tweaking all these microbes and messing with plants and such to suddenly kill you, that seems quite irrational whenever you actually look at what's happened with society. And so really what's going on here is pretty simple. People fear what they don't understand. And rather than ask questions and learn, people will just run with BS and make stuff up on the fly to create a situation that creates fear. And that's unjust and unfair. So one thing I'm going to push you as a student here is to not be afraid, to actually ask questions and try to learn. This is crucial. Failures to do so lead things to like anti-vaxxers. So the reality is we need to talk, we need to discuss, and feel free to ask questions. Again, we'll have time in our interactions in the classroom to talk about these things. So one other thing I want to point out of how we're using genetic modified organisms or recombinant DNA technologies in something called bioremediation. Bioremediation is a cool newer topic where we can actually use microbes to clean up messes we've made as humans. So instead of causing the oil to slip to the bottom of the ocean where it would eventually be broken down in an anaerobic environment, why not use genetically modified organisms to actually break it down on the surface and get rid of it before it ever interacts with other things? And so the idea here is we can actually design microorganisms to do very important tasks for us, including clean up the environment. So people have an unnatural fear of things like bacteria and what many people call germs and such. And again, I say irrational because the fact is anything that can make you sick we call a pathogen. But whenever we talk about pathogens, that includes things like viruses, bacteria, fungi, protozoa, or even helminths. But really, of all the different species that exist, we've only really found 2,000 different microbes that can cause disease. That is a fraction of what's out there. So the reality is most things, most microbes, aren't going to make you sick and are typically good for you and healthy. And so the reality is whenever you kill 99% of what's on your table, you're only killing a small percentage of actual pathogens. The rest are actually helpful things for us. So again, perspective is something that we're going to work on in here and something that you really need to pay attention to as we go on. So to illustrate this, let's mess with your perspective for a minute. I want you to focus on the left side here on the causes of death in the United States. So here's our top 10 causes of death. Another thing I want you to note is in red, we see things that are considered to be caused by microbes. So influenza and pneumonia is caused by microorganisms and can kill you. But notice where it ranks on this list. All the way down here, number 8. Number 8. You're far more likely to die of heart disease, cancer, chronic lower respiratory disease, accidents, stroke, Alzheimer's, and diabetes. But do you think microbes are causing heart disease? Do you think microbes are causing your accidents? And the answer is no. You're terrified of interacting with something that can cause influenza or pneumonia, but you'll go eat that cheeseburger that's going to cause heart disease. So again, perspective is important here. In the U.S., infection is a very, very minimal killer here. And it's important for people to understand that we're far more likely to die just from in your trip coming to class than you are to die of infection. So again, the fear of infection is somewhat irrational. But I want to show you something else. Notice the right side that talks about the number 1 through 10 causes of death in the world. If you notice here, there's far more red things, like here, HIV and AIDS and diarrhea, because again, there are big differences between us and the rest of the world. Infection becomes far more serious in the rest of the world because you have to understand most of the undeveloped world does not have medical practices. They don't have lots of medicines readily available, and thus people are far more likely to get sick and die. Notice number 7 in terms of the world killer is diarrhea. Upwards of 3 million children a year can die of something as simple as diarrhea. That is as much as HIV and AIDS, and that's a terrifying notion. Again, we don't fear diarrhea here, and it's because we have healthcare systems, we have clean drinking water, we have, again, sources of sewage treatment. Again, if we were to have clean drinking water in the rest of the world, we wouldn't see these number of cases. So perspective is important here and something that we're going to mess with purposely with you all so that you understand really what's at risk and how you can protect yourselves. So if you look through here, we have again communicable disease, non-communicable disease, and injuries in comparison to the United States and the world in terms of death rate. And you see again the U.S. is always below the world because again the U.S. has advanced techniques in medicine and actually medical facilities pretty commonly found throughout the country. We have access to medicines. Again, so fear of death from microbes is pretty unfounded in today's society. Every year, malaria will kill around 440,000 to 700,000 people per year. And it's important to understand that that's a huge number. That's more people than die of really infection in this country easily. And this is just one disease found in the undeveloped world. And so it's important to understand that really perspective is important. We don't hear about malaria here frequently because we don't have it. But in the rest of the world, malaria is somewhat common, especially around the equator, and can actually kill lots of people. Malaria is actually a protozoan disease spread via mosquitoes and can be prevented if you prevent the mosquitoes from interacting with people. So really, we can eliminate this vast majority of deaths by just making sure people aren't interacting with mosquitoes. So even something as simple as a bed net could actually save hundreds of thousands of lives each year, which could cost as little as $5. And so the idea here is families can't even afford a bed net to protect themselves. And this is really what I'm stressing about the difference between the United States and the rest of the world. It is not fair in any way. The undeveloped or underdeveloped world live in situations where they must fear disease. While here, you go spend $5 on a bottle of Lysol that you readily spray on your counter for fear of the 0.1% of things that could actually make you sick there. But the reality is there will always be new and emerging diseases. Again, viruses actually change at a pretty high frequency, and as a result, viral diseases will be more commonly found than other diseases like bacterial or fungal. But the idea, though, is there's always going to be something new. And as such, we can try to prepare as much as we can, but until we see the new thing, we don't really know what it is. Where things get more concerning is what we call re-emerging disease, or diseases that we thought we had controlled. Diseases we thought that we knew how to protect people, but yet it's still going on. A good example is things like mumps or measles. In fact, we've seen a huge spike in measles in the last really 4 or 5 years in the United States. Measles was largely eradicated for a very long time in this country, but due to failures in vaccination, measles is back and killing people. People fail to realize that measles will kill you. And thus, whenever they came out with the measles vaccine, people lined up to get it. Nowadays, people are voluntarily choosing not to get the vaccine and thus putting themselves and others at risk. I don't like doing a lot of rants, but this is an important one to kind of bring up here. The reality is the anti-vaxxer movement started whenever, again, a published paper suggested there was some kind of link to vaccines and autism. And so people started failing to get vaccines. And so today we're starting to see the ramifications for this. People fail to remember that vaccines save lives. It should be simple. Get the vaccine, don't get the disease. But again, in today's society, there are enough people that are not vaccinated that the disease has somewhere to live, and thus people can routinely get it and die. I want to point something out. People are quick to say, well, it's my choice. The reality is it's not your choice. The fact is a large proportion, more than you would actually think of, cannot get vaccines due to things like allergic response or even underlying health conditions like cancers and such, that even though they would love to have a vaccine and protect themselves from measles, they can't. And so in order to be protective, they rely on you to make sure that you get your vaccine to protect them. It's something called herd immunity, and actually incredibly important. You see it in nature. Again, the biggest buffalo circle the weakest buffalo, trying to protect them from the predators. That's the whole idea here. If you get the vaccine, you should get it so that the people that can't get it are protected as well. And so whenever someone chooses not to get the vaccine, they're not just putting their lives at risk, they're also putting these people in the weakest situations at risk. And that is, again, an unfair choice that people must rationalize whenever they decide to fail to get a vaccine. Really, we talked about the idea that there will always be new infections or diseases, but another thing we should probably point out is there will be some diseases that we recognize today as actually now being caused by a pathogen. A good example is gastric ulcers. For a very, very, very long time, we considered that gastric ulcers were caused by stress, which isn't completely wrong. But nowadays, we recognize it's actually caused by a bacterium called Helicobacter pylori. And we actually owe this to a physician who was looking at ulcers. And he kept noticing the same bacteria showing up in the vast majority of ulcers, H. pylori. And so to prove that he actually thought that H. pylori had a role, he infected himself purposely with it and documented his progression and formation of ulcers. And thus he really proved that gastric ulcers could and will be caused by Helicobacter pylori. Another thing that's really changed in recent history has been this understanding that many cancers may actually be connected to bacterial and viral infections, that microbes can actually interact with our DNA and promote infection, but also promote cancer. And so we're starting to recognize that really some forms of cancer may be directly tied to infection. And as a result, this is a good thing because now we can protect against infection through vaccination and thus could decrease rates of cancers. There are some things that are still being learned. Again, there's some minor connection at this point between Coxsackie virus and things with diabetes and schizophrenia. Again, some understanding would go a long way that, again, these may play a role, but maybe not the actual purposeful cause. Again, we're still learning things like MS, OCD, even coronary artery disease, obesity. These things may actually be more commonly connected to infection than we currently recognize them as being. I do want to point out that, again, the Coxsackie stuff, the multiple sclerosis, OCD, all that stuff, still very early in the realm of science and discovery. And so understanding the connections between microbes and these diseases is very important. So do not think that because you have OCD that it was strictly caused by infection. There's probably lots of variables in play here. So we're going to have to talk a little bit about really the progression of history of microbes. And really I want to point out that the first real golden age of microbiology was the connections between obvious diseases and pathogens. Again, today we're discovering many more things that are, again, new. Again, the idea that female infertility could actually be caused in some cases by a chlamydia infection that was left untreated. Again, that liver cancers can be caused by things like hepatitis viruses, or even, again, cervical cancers caused by HPV or human papillomavirus. So, one thing I want to point out is that an increasing amount of patients are going to be at greater risk of infection due to decreases in the immune system. There are lots of different causes, including things like viral infection, like HIV. HIV leads to the inability to fight off infection, and we actually call that now AIDS. Another common thing is things like cancer and cancer treatments lead to a decrease in the immune system and thus an increase in infection. But really, there's an area that's a little more alarming to us, and that's the rapid development of drug-resistant microbes. This is actually much more simple and actually plays a pretty big role in what we had discussed before. This idea that as pressure is applied to something, it either adapts and changes or it dies. And so the overuse of drugs has actually led to that exact scenario, where we force the microbes to either change or die. I want to point out that the change is actually going to be random, but if that random change proves to be a benefit, that through the pressure of applying the drugs, that benefit will be pushed onto future generations. Think of it like this. Say I have one bacteria that is resistant to the antibiotic, and I take the antibiotic. All the other bacteria is going to die, and what's left is going to be that one bacteria, right? But now that one bacteria that was resistant to the drug has all the space and all the resources. So what's it going to do? Well, it's going to reproduce, and thus it's going to become more common. You compound that over time, and now you can understand how really evolution starts to happen. How now this thing that has an advantage is going to outcompete everything else as long as the pressure is still applied. So let's talk about some characteristics of prokaryotes. Again, whenever I say prokaryote, I'm talking about bacteria and archaea. But again, they're similar to one another. That's why we can group them together to prokaryotes. But overall, in comparison to eukaryotes, prokaryotes are roughly 10 times the size of eukaryotic cells. So again, it takes about 10 bacteria to equal roughly the size of one of your cells. Another important difference here is prokaryotes do not have traditional organelles. There's no Golgi, there's no endoplasmic reticulum, there's no nucleus. Again, these things are found in eukaryotic organisms. Another important characteristic difference between prokaryotes and eukaryotes is most prokaryotes are what we call single-celled. And so the idea that bacteria and archaea are going to be independent cells. Each cell is its own unique organism. Very important concept. Don't get me wrong, single-celled organisms exist in eukarya, but they're far less frequent. There are single-celled fungi or even single-celled algae. But again, as you're well aware, many eukaryotes are also multicellular. Things like you and I, or plants, or again, animals. So let's show you a comparison. This is not at all a comparison of size. Note, that we're really looking kind of at shape and structures. So notice again we have our eukaryotic cell. It's going to be much larger. It's going to have things like a nucleus where the DNA is in. We have things like mitochondria. We have things like a rough ER or a Golgi. And so the idea here, much more complex. As a simple idea, the bigger the cell is, the more complex it can be. Whenever we look at things like bacteria and archaea, notice it's smaller and thus much more simple. Really about the only thing that we share in common is genetic material, ribosomes, and cell walls and potentially, or cell membranes and potentially cell walls. So let's point out what happens whenever we talk about viruses. If you remember, viruses are considered to be not alive and not even a cell. Because really all a virus is, these are both viruses by the way, really all a virus is genetic material and proteins like capsids. So the reality is viruses are so simple, so primitive, they don't even have the structures we consider to be necessary for a cell. So whenever we talk about the category of helminths, which are worms, this gets to be a weird area, right? Because worms are going to be large and multicellular. But why would we study them in microbiology? Well, it's because they're transmitted like bacterial infections are. And really they start out as a single cell like everything else. And so the reality is they start off microscopic but get big over time. So I want to really stress that since it falls in this weird category of causing infection, but also again being transmitted like infections would be, that makes it something that we would typically look at in microbiology. So notice we're going to do a size comparison here. So as you can see here, we're going to start out focusing on the head of a tapeworm. We call the head of a tapeworm a scolex. And so you can see we're going to really just blow up the surface of the scolex to get to this image. So now just looking at the head of the tapeworm here, we can start comparing things like fungi, protozoan single cells, or even our red blood cells. But if we want to look at bacteria, we actually have to zoom in. So we're just going to zoom in on the surface of the fungi here. And now we can start to compare, using the surface of the fungi, the size of things like bacteria. And you notice this little spot over here, this is a virus. So whenever we look at this, multicellular organisms are going to be big. They're going to be lots of cells. But then as we go to the single cell, now we start to still, in the eukaryotes, see big cells. But by the time we get down to bacteria, they're a fraction of the size of eukaryotes. And then viruses are a fraction of the size of even bacteria. So it's important to remember, again, we study microbiology, the small things. You can't see these with the naked eye. And as a result, you must follow lab procedures with blind faith and try not to screw things up. So let's discuss again the vast majority of microorganisms actually live in places like the outside world. They're typically pretty harmless and often incredibly beneficial. We use them to, produce lots of products. They're crucial to promoting an environment that's favorable for us. But there are some things that actually can cause us problems, and those things we can call parasites. So some pathogens out there actually must live in or on us. Again, that's a parasitic relationship. So, a good example, worms, like the tapeworm we just looked at. The tapeworm is a great example of a parasite. It needs a host to feed off of and actually causes damage while it feeds off the host. So naturally all parasites are called pathogens because they all cause infection. So let's move on now to the scientific method. The scientific method is actually important to all science, but hopefully at this point you've heard it several times. So I'm going to be pretty quick with this. And the scientific method is the general practice of trying to understand natural phenomenon. So I really want to stress that this starts off with an observation. You must observe nature, and doing so you notice phenomena. And so really that can lead to a question. Why is that the way it is? And so what we can do to try to answer said question is to make and form a tentative explanation we call a hypothesis. So a hypothesis is essentially a tentative guess as to what is the cause of this natural phenomenon. So with a hypothesis, it's again an educated guess, if you will. But really, in order to kind of prove or disprove it, we need to gather support. And we do that through things like experimentation. Experimentation gives us data that we can then analyze and use it to either support or refute the hypothesis. I do want to stress that, again, trying to force science to happen, to force a hypothesis into a theory, is not how science should happen. An experiment should be set up to either support or refute it. We shouldn't try to skew the experiment to only support. That's where bad science happens. So again, experimentation is done to either support or refute the hypothesis. And really, the idea is once we get data out, it may or may not support the hypothesis. If it doesn't, we go back and form a new hypothesis and test again until we start to see that correlation between, again, the results and the hypothesis. I want to point out, though, that this isn't where science stops. In order for science to be, again, rigorous, we must test and then retest, and then actually publish and have other people test it. Whenever something is so incredibly supported, we can actually graduate it to a theory. A theory is a collection of statements, propositions, or concepts that explain or accounts for a natural event. But again, the idea here is that this was a hypothesis at one time that has been so supported and replicated, and again, so well approved by the scientific community that we feel like this is a very good understanding of why a natural phenomena occurs. So again, theories do change, but theories are also very well supported. And as a result, theories should be taken pretty seriously. And they're not perfect. Only whenever we think a theory is perfect do we graduate it to a law. As I said before, again, laws are as close to perfect as we can get. And there are really none of them that exist in biology. So really we just have our major theories, like again, cell theory, genetics, homeostasis, ecosystems, and evolution. Science, again, is going to be a progressive thing. There will always be change as, again, there are new instruments and new abilities to test through new experiments. Again, some old theories may be re-examined and altered as time goes on. Again, theories are not absolutely perfect. It's the closest thing to perfection we have, again, are laws. So there are some characteristics that would naturally make up a better scientist than some other characteristics. Things like curiosity, open-mindedness, skepticism, creativity, cooperation. The reality is we must be able to find natural phenomenon, be creative in trying to understand why something happens, develop hypotheses that can be tested and then retested through cooperation. The reality is, as a scientist should, again, have faith in what has been proven again and again, but should always kind of question and always push to find and discover the truth. And so the reality is things will change over time, and that should be how science behaves. Again, everything is subject to change, and as a result, again, science will always change. So I want to point out some examples here. Again, early on, our understanding of how things happen wasn't necessarily great. Again, we thought, spoilage could potentially cause illness. We understood that foods could go bad and make us sick, but we didn't understand why. We thought it was some kind of phenomenon. Honestly, we thought it was probably caused by things like flies, which today, again, sounds crazy, but back then they noticed flies whenever spoilage came about, so that's why they made that connection. Or even things like Black Plague and smallpox was some kind of, transmissible matter. They had this understanding that this could be passed person to person, but didn't know really why or what the source was. So really, again, in the past we had observations, but not really the fundamentals to allow for science to continue. I mean, there was even belief in spontaneous generation in the day. That was this understanding that, again, life could come from nothing. Today, that sounds absurd. We're taught from a very young age that life comes from life, right? Cells divide, and that's how you get more cells. And so back then, though, we didn't have that knowledge. And so it's important to understand that science will change, and that the early stuff seems kind of silly now. But, maybe 100 years from now, the stuff that we're talking about today may be disproven and honestly pretty silly to them. So here's that experiment that was really trying to disprove spontaneous generation. And so, again, Pasteur had hypothesized that there are microbes in the air that could cause that spoilage. And so what he actually did was develop a flask called a swan neck flask, which you can see here, that actually prevented microbes from getting to the broth that has been heat killed. Okay, so we killed all the bacteria in the broth. This broth, free of bacteria, should not spoil as long as microbes does not get to the broth, right? And so he actually proved this by taking and leaving this open to the air, but preventing the microbes from getting in. This broth really didn't spoil, and as a result, he disproved that spontaneous generation. So notice, again, he did leave one open to the air, and guess what happened to that one? That flask, no longer being sterile, was now contaminated with microbes that caused spoilage. So another important person we should talk about is Robert Hooke, and really Antony van Leeuwenhoek. Robert Hooke was the first one to observe microbes, but really von Leeuwenhoek is more important to us because he was the first one to not just observe microbes but start documenting them. And so he actually started with his very primitive microscope drawing the different microbes he saw in the world. And so he was really not just observing but now trying to classify using scientific method. And so really von Leeuwenhoek we actually consider to be the godfather of microbiology, the originator. He really started us off. But thank goodness we have modern microscopes because now we can easily and routinely view microorganisms, again through changes in technology like refined lenses, condensers, changes in focusing, even light sources. So the idea here is as time and technology has advanced, we've changed again our previous understanding. So we're kind of on this understanding that science changes over time. So here's kind of a rough recap of the last 50 years. So again, 70s, we got the discovery of restriction enzymes, or this understanding that DNA can be cut up and manipulated. In the 80s, we actually developed something called PCR, polymerase chain reaction. PCR is actually incredibly important because it allows us to copy genetic material outside of a cell. So this is the first time that really genetics could be done on a wide scale level. And we started to realize that some of the DNA we thought was junk actually played a role in production of what we call small RNAs and their ability to manipulate what genes were expressed. Or even in the 2010s, the big project here is the microbiome project, this understanding that microbes live in and on us and actually play a role in our health. So the idea here is science is ever evolving and will always change as technology and again understanding advance. So with some of these early things that we're going to discuss here, do understand again through today's lens it looks silly, but back in the day the discovery that there were microbes in the air and dust was actually a big idea. That really microbes are found everywhere was a groundbreaking discovery. Today we take that as common knowledge, but back then it was revolutionary. So as I was stressing again, John Tyndall's discovery of microbes in the air and dust that are heat resistant doesn't sound that interesting today, but back then was a huge development that saved lots of lives. Or even Cowan that really discovered that there are endospores that are highly resistant, but if we kill them, we can assume everything else is devoid of life, which we now refer to as being sterile. And so the idea here is these early advances don't seem that exciting for us today, but really paved the way for today's modern advances. Koch and Koch's postulate that proved a specific organism caused disease doesn't sound that exciting today because we know most of the causes of many of the diseases out there, but back then this was literally groundbreaking. Or Wendell Holmes and Semmelweis discovering that really you can spread infection from one place to another, like moving microbes from the autopsy room to the delivery ward. Or Lister understanding that you should wash your hands before you cut into somebody was a big groundbreaking development. So Pasteur is somebody I do want to spend a little more time talking about here. Pasteur did a lot for microbiology. Pasteur really developed a lot of modern techniques and as such should be very important to us. He invented things like pasteurization, which you're probably well aware of. But he also developed things like a lot of lab equipment we use today. He also developed things like, again, a lot of techniques for manipulating microbes. He developed many vaccines. He discovered lots of pathogens. So Pasteur is probably one of the most influential people in the history of microbiology. Robert Koch, as we said before, very important for the idea of proving disease through establishing and verifying germ theory. And so again, Koch's postulates very helpful to us in understanding disease transmission. But one of the last things we need to do here, and I understand this has been kind of a crazy rambling, but that's what really the introduction chapters are. One of the last things we need to do here is talk about nomenclature, or the naming system that we use. We use in biology a binomial nomenclature, which means we use the last two levels of classification. So for us, that's genus and species. There are some rules here. Every time that we write down a genus, we need to capitalize the genus. So look down here at Staphylococcus. Staphylococcus is a genus. And notice every time we have the word Staphylococcus, we're going to capitalize it, illustrating that it is a genus, because genuses or genera are always capitalized. Species names, on the other hand, should always be lowercase, like Aureus down here. Notice again, Aureus, little a. So it's important. So whenever I see Staphylococcus aureus, I understand that it's part of the genus Staphylococcus, but belongs to the unique species Aureus. So actually, after I've written or typed this the first time, I don't have to keep writing out the genus. I can just abbreviate it, but notice the abbreviation, it's still capital S here. Okay? But I'll always make sure and record the species if it exists. So, in order to illustrate a scientific name as a genus and species, again, capitalize the genus, lowercase the species, but it's also important that if you're handwriting it, to underline it, telling us it's a scientific name, or if you're typing it, like here, notice it's italicized. So italicize it or underline it, and then capitalize genus, lowercase species. That way we're all on the same page that we're working with a organism. So some things to talk about with classification, identification. We as humans have always wanted to classify and organize everything in existence, but the reality is it's super hard to do. The fact is that we in the past used to classify things based off of specific features, but nowadays we use something called genes and genetics to do it. And thus taxonomy is changing, and actually will be changing for a while. So I'm not going to stress a huge amount about taxonomy with you. The exception, though, is I do want you to know the kind of levels of classification we have: domain, kingdom, phylum, class, order, family, genus, species. The reality is we will discuss things based off of the genus and species name, but I also do expect you to know about the domains and kingdoms. Whenever we talk about domains, we have three domains: bacteria, archaea, and eukarya. So notice in this example of taxonomy, we're looking in the domain eukarya. But an interesting fact about the domain eukarya is we actually have, again, different kingdoms in eukarya, which includes things like plants, animals, protozoa or protista, or even things like our plants. And so the idea here is there are multiple kingdoms: plants, animals, fungi, and protista, which is protozoa and algae. Okay, but the idea, though, is as we go further down, we can specify. So in the kingdom Animalia, we see a phylum called Chordata, which really means you have a spinal cord. And among the things with a spinal cord, we see mammals, which again means you give live birth, you breastfeed, covered in hair in the vast majority of situations. Notice, though, but we also have the order primates, which in primates we have the advanced primates we call Homidae, and then in our genus being Homo and our species being Sapien. So if we were to actually write out a full scientific name, we'd be Animalia, Chordata, Mammalia, Primates, Homidae, Homo sapiens. No one wants to write that all out. Nobody wants to read that all. So again, we use that binomial nomenclature and just call ourselves Homo sapiens. That way we know what we are. So taxonomy, as I said before, is going to be changed, will change. Classification systems are still in flux and changing. So this understanding that, again, science is always changing, and really taxonomy is no different, is important for us. Again, phylogeny was based on evolution, but again has been changing over time. Notice in the past we used to consider really two domains, prokaryotes and eukaryotes, but since then we discovered that we should probably break prokaryotes up into archaea and bacteria. So again, we have the domains bacteria, archaea, and eukaryotes, which we can see here, right? So bacteria, archaea, and eukaryotes. Notice we don't have kingdoms under bacteria and archaea. They don't really exist nowadays because of that break, but notice that we do have our kingdoms of eukarya, which include plants, animals, fungi, and protista.